So today I will talk about a project, an ongoing project that I'm doing here at NYU Abu Dhabi in the lab of Dr. Claude Desplan. And so the title of my talk is the developmental patterns of the Drosophila visual projection neurons. So we all know that the brain is the most complex organ, and this complexity is largely due to the vast diversity of neurons that process different types of information and perform different functions. So one of the main questions and goals of neuroscience is to try to understand how we generate this vast diversity of neurons. So in our lab, we use the fruit fly as a model organism, and more specifically, we use the visual system to try to understand a lot of these questions. And so the visual system of the fruit fly, or the optic lobes, it's made up of different compartments or neural pills, and each one is essential for processing different kinds of visual information. So briefly, visual signals are first detected by photoreceptor cells in the retina. Then that information is then sent to neurons within the lamina and the medulla. In turn, medulla neuron then send that information to the lobular complex, which is made up of the lobula and the lobular plate. And lastly, medulla, lobula, and lobular plate neurons then send that information to the central brain for further processing. And so despite its small size, the optic lobe of the fruit fly is very complex and contains a large diversity of almost 200 subtypes of neurons that has different morphological and functional characteristics. And so a lot of work and effort had been put forth to try to understand how we generate this diversity of neurons in the optic lobes of the fruit fly. And so it was found that fruit fly neurons originate from two main neuroepithelial domains. The first one is called the outer proliferation center, or the OPC, and the inner proliferation center, and IPC. And these domains are further divided into compartments or subdomains called the main and tips of OPC and dorsal and ventral IPC. And throughout this talk, I will touch more into these different domains. And so from the OPC, retinal cells or optic lobe neurons are generated via two different mechanisms. So the first mechanism is called temporal patterning. So we have these neural stem cells or neuroprogenitor cells. They express distinct transcription factors, and these then divide to form ganglion mother cells, and then which further divide to form different neurons. And then as time progresses, these neuroblasts or neuroprogenitors express a series of temporal transcription factors, generating at each time point a different neuronal population, or various neuronal populations. Additionally, the OPC is further divided spatially into different compartments that also express different factors. And so the combination of both temporal and spatial patterning is what allows for the generation of the vast diversity of neurons that we see in the optic lobe. And just going back to this figure that I showed previously, and so the majority of the studies that has been done before on trying to understand the diversity of neurons in the fruit fly optic lobes have been done on the medulla, which is the largest neuropil. But also a lot of work has been done on the lamina and lobular plate. However, the lobula, which is an essential component of the circuitry, the neurogenesis of its neuron is still very poorly understood. And so I wanted to study the lobula and focus on a subtype of neurons within the lobula called the lobular columnar neurons, or LCN, or LCs as I will call them from now on. So LCN neurons, they are projection neurons, meaning that they receive information from the medulla and lobular plate, but they have axons that project out of the optic lobe and into the central brain for further processing of visual information. And so you see these neurons have processes in the lobula and axons that go out to the central brain. And so lobular columnar neurons, there are about 20 different subtypes of these cells. And they have distinct morphological pattern and projection patterns in the central brain. And so Jerry Rubin lab from Janelia, they recently published these transgenic fly lines, and within each there's an expression of a specific subtype of lobular columnar neuron. And what's interesting is I mentioned there are about 20 different subtypes, but about actually half of them were only discovered recently in 2016. So really not much is known about these cells. And what's very also interesting about these cells is that when they project into the central brain, they do so in a very cell type specific manner, meaning that each one of these lobular columnar neurons, they project to the central brain, forming these highly specific neighboring synaptic domains. And so each one of these colors represents a termination zone of one of these different LCs. And so, you know, they have very unique projection pattern in the central brain, and within the lobula itself, they also have a unique stratification pattern, dendritic pattern within the lobula. So the lobula can be subdivided into 6 layers, and each one of these lobular columnar neurons project to a distinct layer within the lobula. So each one is very unique in terms of their morphology. And as I mentioned before, a lot of these neurons were only recently discovered, so a lot of work and effort now is being put towards trying to understand the function of these neurons. And as I mentioned, these neurons receive visual information and then send that information to the brain for further processing. And so these neurons basically control visually guided behaviors. So for example, here, this LC6 neurons, they respond to looming stimulus. And a looming stimulus is basically it mimics the approach of a predator from above. So imagine you see a fly and you try to hit it, the fly immediately flies off. And so these neurons basically control these behaviors. As you can see here, when these LC6 neurons are activated, the fly basically has this takeoff behavior, similar to when they are activating by looming stimulus. So basically, these neurons alert the fly that there's a predator or a dangerous signal, and it tells it to fly away. So as you can imagine, these neurons are very essential for the survival of the fly. And so other LCs, the functions, so LC16, they also, they're activated by looming. However, they have a different behavioral outputs. So when LC16 neurons are activated, the fly actually they basically have a retreat. So they walk backwards, turn around, and fly away. So 2 neurons, 2 LCs respond to looming stimulus, but with very different behavioral outputs. And LC10 neurons were found to be important for courtship behavior and tracking flies. And LC11, for example, are essential for detecting small moving objects. So like I mentioned, these neurons are very essential for the survival of the flies. And like I mentioned also before, a lot of work is being done now to try to understand the development of these neurons. However, when it comes to their development and neurogenesis, very little is known about these subtypes. So for today's talk, I will focus more on the neurogenesis aspect of these neurons. And if you're interested in the development, I have a poster tomorrow which talks more about the development of these neurons. But so we will talk about their neurogenesis. And so here, this is a schematic that I will go in detail with, because I will be using this schematic throughout my talk. So this is a schematic of an adult fruit fly brain, and you see on this side we have the optic lobe with the different neuropils, the medulla, lobula, and lobular plate. And on this side we have the central brain. And you can see here, these are the different colors represent the different optic glomeruli, and each one of these circles here represents a different subtype of lobular columnar neuron. And these dotted lines here represent LCs that are located posteriorly. So these, for example, here, LC11 and 21 are located behind LC6 and LC9. And so as you can see from the schematic, lobular columnar neurons are situated at this interface between the optic lobe and the central brain. And due to that position, it was very ambiguous as to where these neurons are coming from. So do they have an optic lobe origin, similar to other optic lobe neurons, or do they have a central brain origin? So the question came, so where do LCNs come from? Where do these neurons come from? And to try to understand this question, we employed various techniques. And so we used single-cell RNA sequencing, and we used these split Galford lines, the specific lines that are specific for each one of these different LCN subtypes. And we tried to look for markers and genes that are expressed in these cells. And as I mentioned previously, as many of these neurons were only recently discovered, not much is known about the types of genes that they express. And so we tried to annotate the LC clusters. And so our lab, as a collaboration between here and New York, we performed single-cell sequencing of all optic lobe neurons. So we sequenced all of the neurons in the optic lobe, and we were able to basically identify all the clusters for lobular columnar neurons. And so this took a lot of time and effort because, as I mentioned, not much is known about these cells. But we were able to annotate all the clusters, which allowed us to really have a better understanding of the transcriptome of these cells. And what's also really good about this data set is that the single-cell sequencing was done not only in the adult fly, but we have it throughout at different developmental stages, from early pupil stages all the way to the adults. So we can follow them developmentally at different time points. And so going back to this schematic, and so first we wanted, as I mentioned, we wanted to know where these LCs come from. And the first hint that we had for an origin of lobular columnar neurons was for LC6. These LC6 neurons that here project to the central brain. And Claire Bertet from our lab showed that these neurons originate from this domain, the tips of the OPC. And within the tips of the OPC, neuroprogenitors also undergo a series of temporal transcription factors. And again, at each time point, there are different neuronal populations that are born. And so she found that these LC6 neurons are born from this time window, the sloppy paired time window, where the neuroprogenitors express the transcription factor sloppy paired. And these neurons express a transcription factor called TOI. So we wanted to know whether other LCs also come from that domain. And so here we looked at LC9. So LC9 neurons, they are situated next to LC6. They follow the same trajectory into the central brain, and their optic glomeruli are neighboring. So could they also be coming from the same domain? And so when we looked at the transcriptome of both LC6 and LC9, we saw that they cluster very close together, meaning that they share a lot of common markers. And so this was a hint that maybe they are indeed coming from the same neuroprogenitor domain. And so we looked at some of the markers that LC9 neurons express, and one of these markers is TOI, which, as you remember, this is a marker of neurons that come from that sloppy paired time window. And you can see here LC9 neurons, they express TOI similar to LC6, which are neighboring them. And so to further confirm that they do in fact come from that sloppy paired time window, we performed an experiment which we call FlexAmp. And so in this experiment, we have a way of labeling neurons that are born at a specific time point. And this allows us also to label them and maintain the expression throughout the adult stage, which can give us a chance to really understand their morphology and identify the neurons. So we did this experiment in a line that is expressed in sloppy paired positive progenitors. And when we looked in the adult, we found that we indeed find both LC6 and LC9, meaning that these neurons come from that domain. And here you can see the LC9 and LC6 optic glomeruli are neighboring. And here these are the cell bodies. And again, they follow the same trajectory into the central brain. And so we concluded that both LC6 and LC9 come from this TOPC domain of the optic lobe. And so what about other LCs? And so if you recall previously, I told you that optic lobe neuron originates from these two neuroepithelial domains. And this particular subdomain, the ventral tip of the IPC, Philippe, who is here, and when he was in our lab, he showed that the ventral tip of the IPC actually doesn't undergo temporal patterning, but rather there is spatial patterning. And so we can subdivide the ventral tip of the IPC into three regional domains. And within each domain, the neuroblasts divide and give rise to three different neuronal populations that also express different transcription factors. So for example, region 1 of the VTIPC gives rise to neuron that express the transcription factor Rx. Region 2 gives rise to neuron that express ACG6 and TOI, while region 3 gives rise to neurons that express Rx and TOI. And so we wanted to see whether any LCs come from that domain. And so we performed the FlexAmp experiment again in a line that is expressed in the three regions of the ventral tip of the IPC. So this is a larval brain, and you see this ventral tip of the IPC domain that is expressed in this line. So we performed the FlexAmp experiment, and we found that we were able to recover different types of lobular columnar neurons. So the first one are these LC12 neurons. So here, this is, again, a section of a fruit fly adult brain, and you can see here, this is the lobula, and this neuron here with the cell body located between the optic lobe and the central brain, they project into the lobula to distinct layers, lobula layers 2 and 4, and they form this optic glomerulus. And so these are characteristics of LC12 neurons. And we also were able to find LC17 neurons. Again, they project to distinct layers within the lobula and the central brain. And lastly, we also found another subtype of lobular columnar neuron, which are these LC4 neurons. And so you can see these LC4, 12, and 17, which seem to be coming from this ventral tip of the IPC. They all have neighboring optic glomeruli. And also, interestingly, when we looked at the transcriptome of LC4, 12, and 17, they also clustered together, which hinted to also the notion that they have a similar origin. And so we wanted to look at the markers that these neurons express to try to pinpoint them to the specific subdomain within the ventral tip of the IPC. And so in this line where we have all three LC4, 12, and 17 expressed, we saw that they express both ACG6 and TOI, which are characteristics of neurons that come from the region 2 domain of the ventral tip of the IPC. And so here now we have these two LC population, one that is coming from the tips of the OPC, and these three neurons, LC4, 12, and 17, again, they have similar cell body location, and their optic glomeruli are neighboring, and they come from a different domain. And so do other LCs come from these two regions, the tips of the OPC and the region 2 of the VTIPC? And to further look at that, we had a line that is expressed only in the TOPC and the VTIPC. And you can see this line here. This is a larval brain again. And you see the green is the expression within here, the tips of the OPC and the VTIPC. And so we performed single-cell sequencing in this line, which would give us information about the types of neurons that are generated from only these two domains. And so we performed single-cell sequencing, and we annotated all the clusters, and we wanted to search for our LC clusters. And as expected, we found the clusters for LC4, 12, and 17. And again, you see these three neurons, they clustered together. And when we looked at the marker, if you remember, I said they express ACG6, and you see this cluster specifically expresses the ACG6 transcription factor, which confirms our previous finding of these neurons coming from that domain. And similarly, we find the clusters for LC6 and LC9. Again, these two neurons cluster together, which points to their common origin. And again, we checked for the marker, as I mentioned previously, TOI, which is a marker for the slot repair time window, and this cluster expresses, which further confirms our previous findings. But interestingly, we also found clusters for other LCs. So here we have LC11 and LC21. And again, if we look back at our schematic, you see that LC11 and LC21 are also located next to each other. They follow the same trajectory to the central brain, and their optic glomeruli are neighboring. And we performed various analyses, but due to the sake of time, I won't have time to go through it. But we found that these neurons do come from the TOPC as well. However, they come from a different temporal window, which is the distalis time window. So they, similar to LC6 and 9, they come from the TOPC, but from a different temporal window. And also, we are able to find the clusters for LC16, 15, 24, 26, and 13. And again, these neurons also cluster together. And again, when we look at the schematic, these neurons are situated close together. And we performed several experiments, and we found that these neurons also come from the TOPC. However, they come from a novel temporal domain. And we are currently analyzing those data to really pinpoint where they come from exactly. And lastly, we found the cluster for LC20 neurons here. And you can see here LC20 are also located close to LC4, 12, and 17, although they have their optic glomerulus is in a different location. And similarly to these ones, to 4, 12, and 17, LC20 also come from the ventral tip of the OPC. However, they come from a different spatial domain. And so, in summary, we saw that lobular columnar neurons, they have very heterogeneous origins. So all these neurons here on this side of the brain, they all come from the TOPC, although they come from very distinct temporal domains. And these neurons on this side, they all come from the VTIPC, but they come from different spatial domains. And so we were left with this population of lobular columnar neurons here that are also located kind of far away from the other two. And we couldn't find these neurons in our VTIPC or TOPC populations. However, when we did more analysis, we found that these neurons actually come from the central brain. So these neurons are not, do not have an optic lobe origin, but yet they come from the central brain. And so this is very interesting and unique because, to our knowledge, it hasn't been shown before that a single subtype of neurons that converge onto a location in the brain to have such heterogeneous and diverse origins. And so in summary, we saw that cell body location and axon trajectory to the central brain as an indication of a common origin, and that LCNs have at least 3 different origins, so they come from central brain, tips of the OPC, ventral tip of the IPC. We also saw that within the VTOPC and the ventral tip of the IPC, that these different LCNs originate from different temporal and spatial domains, and that at least 2 to 6 LCN originate from the same domain. So why do LCNs cluster together? So why are these LCNs, why do they have the same origin but not these ones? So we tried to look at this data in much more detail, and we wanted to see whether it has something to do with the similar functional properties. As I mentioned before, these neurons basically control visually guided behaviors, and so do LCs that cluster together have similar functional characteristics. And so actually it's not the case, because we see that LCs that come from different domains can all respond to the same basically stimulus. So as I mentioned previously, looming stimulus, so we have LC4, they detect the speed of looming stimulus. LP, LC2 here from the central brain, they can detect the size of the looming stimulus. LC6, they also have this takeoff behavior to looming stimulus, while LC16 also can detect looming stimulus. And so it doesn't seem that a common common function means a common neuroprogenitor domain. And also within the same domain, we have LCs that perform different functions. So again, here, LC4, they respond to looming, while LC12, they detect motion onset. LC16, again, respond to looming. LC15, they are activated by moving bars. And LC6 here, again, they respond to looming, while LC9, they have ground discrimination. So what could it be? So could it be something, and could it be some sort of an evolutionary mechanism that allows these different LCs to be grouped together? And this is actually an ongoing analysis. And now that we have the transcriptome of these different LCs, we can try to look for markers that are common between them that could explain this type of organization. And with this, I thank you and thank Claude, and thank Katarina and Raghu for the single-cell sequencing, and Philippe for the VTIPC data, and the lab here in Abu Dhabi and New York. And also, we have our lab posters, so please visit them. And thank you.